Hey everybody, welcome to the video where I do the quiz answers for our lecture on molecular motors and cell motion. So we're going to talk about cell contraction and also cilia and flagella motion that's directed by motor proteins. So the first question is on the movement of cilia and flagella. This is driven by motor proteins, so that's true. All right, myosin motor proteins are responsible for cell contraction in skeletal muscle, cardiac muscle, and smooth muscle. So the correct answer here is all of the above. Remember, skeletal muscle and cardiac muscle have sarcomeres, so they're striated, but smooth muscle also has the that kind of net-like network of actin thin filaments that surrounds the cell and contracts as well. Okay, myosin motor activity is used for muscle contraction and cargo transport. That's important to remember, but it's not used for cilia and flagella motion. Remember, this is the axonemal dynein with microtubules in the cilia and flagella. So the correct answer here is A and B. Next, the drug omecamtiv mecarbil, OM, works by increasing heart contractility. Remember, it does this by pushing the equilibrium in the actin-myosin cross-bridge cycle towards the power stroke side of it. And finally, tropomyosin and the troponin complex regulate myosin activity, sorry for the spelling mistake there, by controlling access to myosin binding sites on the thin filament. Remember, tropomyosin wraps around the thin filament and blocks the sites where the myosin motors bind and can then pull together if we're thinking about, say, the sarcomere, for example. All right, that's the pre-discussion quiz. Let's do the post-discussion quiz now. Which domain is most likely to be conserved across the myosin family? So we had a series of options here. So which domain in myosin is most likely to be conserved? The actin binding domain. So I would think of this as the actin binding surface of myosin, or the motor domain itself, the entire motor domain, or the microtubule binding domain. So this is kind of a non-starter because myosin binds to actin instead of microtubules, or the cargo binding domain. And the key thing here is to remember that there's all these different myosins with different cargo binding domains to bind different cargos. So this is the answer that we wouldn't expect. And then microtubules, this is also another obviously wrong answer. The correct answer that we were looking for is A and B. We would expect the motor domain to be conserved, and also this specific part of the motor domain, the actin binding surface of the motor domain, would also be expected to be conserved. All right, next question. Tropomyosin directly senses intracellular calcium. That is incorrect or false. Remember, tropomyosin is the rope-like filament that wraps around the thin filament. It's the troponin complex, and specifically the troponin C subunit inside the troponin complex that directly senses and binds to intracellular calcium. All right, next question. In skeletal muscle contraction, the binding of ATP to myosin causes relaxation of muscle is the correct answer here. And the way I remember this is I think about rigor mortis. So when people die, their muscles become very stiff. That's called rigor mortis. And the reason for that is because all the cells in their body, and specifically the muscle cells, can't produce ATP anymore. There's no more energy being provided to the muscle, so it can't produce energy anymore. It can't make ATP anymore, so all we're left with is ADP. And in that state, the muscles, actually the myosin in the thick filament, the myosin heads bind to the thin filament and cause the muscle to stiffen up. So that's how and when ATP binds during normal, say, muscle contraction during the actin-myosin cross-bridge cycle, this causes the release of the thin filament and gets it ready to do the cocking motion and then eventually the power stroke at the end. All right, after hydrolyzing ATP. All right, next question. The relationship between microfilaments and myosin is the same as the relationship between microtubules and so obviously myosin is the motor protein on actin microfilaments. So on microtubules, we have two motor proteins, dynein and kinesin. So these are both potentially correct. You could say these are both reasonable answers, but a better answer is kinesin, because myosin is generally plus end directed, and so is kinesin. All right, so that's why something to think about when you're taking the exams is sometimes we will include multiple answers that are okay, but one that's clearly better. And this is an example of that, where kinesin is the answer we were looking for. All right. And final question. track switching from myosin, actin to dynein microtubules would be most likely to occur for the answer we were looking for here is an endosome. A lot of you said secretory vesicles. So this is certainly there's track switching for secretory vesicles as they go from, say, the Golgi to the plasma membrane. But the key here is going from myosin actin to dynein microtubules. So this implies going from the outer cortex, where we have this thick actin filament network right underneath the plasma membrane, to more internal into the cell, to the microtubules, which are more going to, say, the centrosome microtubule organizing center, going back towards the say the Golgi and the ER. So if we're going in that direction, in a retrograde direction, that's what we'd expect to see with this type of track switching. So that's going to be an endosome, a recently endocytosed vesicle. This is going to be moving in the opposite direction to a secretory vesicle. So that's the correct answer we're looking for here. Okay, that's all for these quizzes. Good luck studying for the exam, everybody. And I will, if you're watching through all of these, I will see you on the next video. All right, I'll stop the recording now. Okay, see you next time.